Okay, we're recording now.
Excellent. All right. Welcome to our, I don't know how many umpteenth journal club meeting we're having here. So the paper I picked was the Parental Olfactory Experience Influences Behavior and Neural Structure in Subsequent Generations, right? That's the one everybody read, I hope. It's a Nature Neuroscience paper. It's got some heavy-duty, I guess, implications. And I'd like to, I guess we can dive into that and talk about those implications later. But first I want to do a little bit of background on what this paper is about. It's an epigenetics paper. Yeah, it's an epigenetics paper. So I don't know, does everybody see these slides that I have up here? Yeah? Okay. Yeah. So, all right, so the first slide that you're looking at here is actually a figure from a review paper that I just sent out, a News and Views article that came out recently, January 2014, Nature Neuroscience. So it's got a, title where they say Lamarck Revisited, and this image just lays out the groundwork for the actual experiment that was performed. So essentially what this paper is all about is that they're purporting that you have a fear-conditioned response being transmitted through generations, not just the F1 generation, the first generation, but also the second generation, the F2 generation. And what they're looking at here is basically mice. So they've taken mice and so they've taken specifically, they used both female mice and male mice. And a lot of the work that they've done in this first, the paper that they show here, but they also have supplemental data, is focusing on the male mice. So the fear response is basically a conditioned response to an odorant, which is this acetophenone, which has a smell of cherries or almonds, coupled to a shock. So after they do this to these poor mice, these F0 mice, which is the parental mice, they have a fear response to this smell. And then they cross these mice with and there's a reason why they pick this particular smell. It's because this odorant has been worked on before in the past, and so the pathways that are characterized pretty well. So that allows them to do a lot of assays. So what they specifically, the odorant acetophenone binds to are these odorant receptors called, they're found in M71, I believe, they're found in cells in the olfactory bulb. It's these OLFR151 genes that encode these M71 receptors that bind to acetophenone. Hello? Yeah, so it's a very specific detection for this smell, and the neurological how the neurons are basically laid out has been mapped very well also. So they can then take a look at and see what happens to the glomeruli, which are these this region in the olfactory bulb where these neurons basically sprout and make connections. So they have this kind of this really nice model here where they can do a lot of assays, and they can ask a lot of questions and get a lot of answers back after they do this conditioning response. And that's why they pick the acetophenone and this olfactory system to do the conditioning versus some other type of I guess conditioned response. So the gist of this paper, just to give a little background before I go into it, is that it seems, at least they're claiming that this fear response is propagated to the F1 and F2 generations, and we'll go into the kind of the methodology behind how they've done this. But it seems that the mice in later generations have an imprint of this smell. So I want to revisit that in a little while, but first I want to talk a little bit about the implications as to how this could be transmitted. I think these are a little out of order, maybe. Let me go to the next one. So this picture here, this is, so epigenetics, just to give a background, is a phrase coined by Conrad Waddington back in like 1940s, really before we knew what the structure of DNA looked like. And he basically there's this image which has been really is held fast pretty well. It's this marble rolling down this landscape, this epigenetic landscape. And this epigenetic landscape basically influences the way, so the low part of where the marble ends up is essentially the phenotype that's being expressed. So as this marble kind of ping-pongs down, the differentiation pattern of cells gets influenced during development by a variety of factors, and the cells are then locked into a certain position. So this phenomena was first proposed by Waddington before anybody really knew what the chemical mechanisms of this were. It was basically a very phenomenological description, but it's a very elegant design. And since then, we've sort of been able to tease out what is happening in a lot of cases. So epigenetics, just to give a quick description or definition, it's essentially modifications of the genome that are inherited without altering the sequence of the underlying genome. So that's a pretty big, definition. There could be a lot of things that'll constitute epigenetic changes. Some of them are much more characterized than others. So I'm just gonna tag up the original image of Waddington here, Conrad Waddington, and add in methyl groups. So I kind of imagine methyl groups. So methylation is this little chemical that can be attached to both cytosine residues directly on the DNA itself, or even on the proteins that the DNA coils around, which are the histones. And depending on where these methyl groups go, either on the DNA, where it can actually act as a repressive function, basically shutting down gene transcription, or it could basically methylate histones, and depending on where on the histones it methylates, it could have functions related to methylation or activation. So I'm kind of drawing these methyl groups here as blocking potential conduits to gene expression to kind of guide the marble along to where it eventually ends up. So you can sort of imagine the underlying landscape as the sequence. Since then, more modifications have been determined. So we've got acetyl groups. So the acetylation status of histones basically opens up chromatin, so it allows expression to happen. So it sort of has the opposite of effect of this methylation that happens on the cytosines, which happen in these regions, which are called CPG islands, the P basically standing for phosphate. So when you have a lot of C's and G's, you have this methylation happening in mammalian cells to a great extent. So those are just two of the better characterized modifications. I was recently, I don't work on the epigenome personally. I remember somebody telling me that there are literally dozens of other little chemical groups that can be attached to DNA and histones that are still being worked out as to what function they have. So it's a pretty broad landscape. And I have a few slides here showing the kind of methodology you can do to at least one technique out of many that you can use to read the epigenome. And this is a technique that was used in the paper, which is called bisulfite sequencing, where you chemically treat the DNA with this compound bisulfite, and it'll actually convert the cytosines into uracils. So clearly, if you add in bisulfite, you get the cytosines converted to uracils, then you could sequence that and find out what used to be a cytosine. However, if it's methylated, then what happens is that the methylation protects these cytosines, and then they remain cytosines. So you can then indirectly determine whether or not this region has been methylated or not. So that's one technique in that they employ in the paper. They use other techniques using antibodies to certain regions that have been modified to do an affinity, do immunoprecipitation. But this is one technique that they mentioned, so I wanted to kind of briefly go over that. So that's kind of my background. So are there any questions before we kind of start going into the paper? No?
One question I had real quick on the CPG. Is it the case that methylation always occurs when there's the C next to the G?
Well, that's my understanding. They refer to them as CPG islands, where you have a kind of a high density of these C's and G's, but I don't know the details as to what particular order it has to be, or what the density level it has to be. Can it just be 1 cg, or can it be like 100? I'm not sure.
Is the P phosphate in that CPG? Is that what?
Yeah, that's a phosphate. Yeah.
So I assume it means that it's adjacent CGs, not kind of across the ladder CGs, right?
Yeah, they're adjacent. That P just means phosphodiester backbone.
Right. Okay.
That's my understanding. So they could have just called it CG.
Yep. Okay. Thanks.
Yeah, I guess, and that does mean adjacent. So yeah.
Yes. Yeah. So so this paper has a number of different terms, and I'm not really familiar with the methodology behind doing these types of behavioral assays. And as I go over this paper, I'm going to raise a couple of issues, because I read this paper once and then read it again briefly again, and I'm going to go over the experiments, but I want to ask as we go along whether or not, I thought when I read this paper that there was maybe one major control that was left out, but maybe they actually did it and I didn't read it properly. So I want to address that when we get to the first figure. So I laid the groundwork a little bit as to why they set up the experiment, why they chose this particular receptor, because it was really well known. So they used two different types of mice. They used these C57 black 6J mice, which are a common mouse, I think, used in studies. And they used another mouse called M71 LAC-Z, which was a transgenic mouse. And the reason they used this mouse is that it expresses beta-galactosidase in these M71 cells in the olfactory bulb. So they can actually stain these cells and find out what's happening morphologically or anatomically in these mice, what the difference is anatomically later, just by staining that region with the appropriate substrate, and that's what they do in this paper. So basically this paper consists of looking at these really kind of three major sets of experiments, a lot of behavioral experiments, a lot of looking at the morphology of this glomeruli to see how it changes in response, how it changes. And also, they finally look at do a little bit of biochemistry with this bisulfite sequencing to see, to try to, posit a mechanism of action. Although there's a lot of details left out. So they say in this paper they use this olfactory fear conditioning. So the first thing they do is with the adult mice, they do, if in the supplemental, and I don't have it out, they kind of have a nice schematic of stepwise how they do this. They do something called a fear-potentiated startle with the F0 mice, which is the initially the male mice. And what they do is they kind of electroshock the foot pads of the mice, and they subject them to this odorant, this acetophenone, and they try to condition the mice to associate the smell with a bad feeling, which is this electroshock. And then they want to see if this conditioned response is propagated to future generations. And they take a lot of pains here to try to make sure that this is actually biologically transmitted, that it's not that it's genetically transmitted, and that's not something that's a learned behavior. So they go out to the F2 generation, which is 2 generations out. They also rear the mice using surrogate parents. They also do in vitro fertilization just to make sure that maybe acetophenone-treated F0 male mice are particularly aggressive and the females don't like them, and maybe that somehow is influencing the pups as they're being gestating. So they do all these controls to absolutely determine that this phenomena is being transmitted genetically and is not being conditioned somehow into the pups as they're in the cage. Okay, so that's how they do the conditioning, and they have several different terms here. They have F0 home, F0 ace, and F0 prop, and it's used throughout the paper. And for the future generations, it's respectively F1 and F2, so that you'll just see F1 home, F1 ace, F1 prop, and the same thing for F2. And home means basically that the mice have not been conditioned; they haven't had a smell given to them. F0 ace means that they've been conditioned with the acetophenone, and F0 prop means that they've been conditioned with another chemical, propanol. So basically, this other chemical has nothing to do with ACE, and the reason they pick it is that it goes through a different pathway. It uses a different, not OLFR 151, but I believe OLFR 6 or something like that. So different receptor for it. So they have a control for that. And they say also that they didn't exclude any mouse from the study. So everything was used. It was double-blinded. Okay, so then what did they do? So the one interesting thing, the one caveat I had was initially when they have the F1 mice, this first generation mice, what they do is they do these measurements using something called, and I don't really know the details of how it was really performed. It was called an odor potentiated startle. And one thing they mentioned about this odor potentiated startle is that there's a caveat here to this, is that I'm trying to find the sentence here on page 2, is enhanced. So they don't look at these assays that they do. They say they don't look at the valence of the stimulus, the valence of the valence specificity and the emotional value of the odor. So what that means is that these mice are sensitive, but these assays they use are, they say enhanced OPS to acetophenone in our experiment would be interpreted as an enhanced behavioral sensitivity to acetophenone, not necessarily an increase in fear to acetophenone. So I'm not sure if that, that doesn't seem like it really strengthens their, initial argument that this is a real conditioned fear response. And one other issue I had before I go further is they, and I don't know, I'd like to hear people weigh in on this, is that before I go into figure 1 and talk about the behavioral sensitivity, and it certainly suggests that these F1 mice are behaviorally more sensitized to the smell, is whether or not they controlled for in F0, where acetophenone is introduced without the conditioning shock, right? So, so everything that they've done, they've shocked the mice and they've introduced acetophenone, but I don't know if they actually just had the smell without the conditioning. And the reason why it seems to me kind of important is, so first of all, they use this OPS test, this odor potentiated startle, which doesn't distinguish between a fear response, I think, and general sensitivity. And also, they cite a study in the back of the paper, which is reference 37, that demonstrates that there are heritable effects of sensitivity to acetophenone if you just put it in the diet of the mice. So it's known that the mice get sensitized to acetophenone and it's transmitted. But whether or not it's an actually